Today, not doing any particular job. What I thought we would have a look behind the scenes of a publication which we published this year or recently. Let's look. We'll have a quick review of this and some of the code behind it, and what went into some of the processing and the analysis and printing the figures. So we'll have a quick browse. So a pipeline, as always, building these, and this is applied to the honeybee nests, microbiomes in these. One thing that struck me from studying microbiomes is it's considerably easier than trying to do metabolic coding for plant for higher taxa insect communities, and a lot of the methods are built to primarily on this system. So on 16S, it's very easy to get high quality data, so you can consider that if you're looking for somewhat of an easier domain to work on, more easier job of getting reliable sequence data. Anyway, so here's an overview of the kind of the pipeline, sampling bee nests, metabolic coding. We use ASVs. These are amplicon sequence variants, kind of shifting away from OTUs in microbes, so a bit more high resolution. But this gives us directly composition. Then phylogeny from 16S gives us phylogenetic diversity. Use a green genes database, taxonomic diversity, and KEGG gives us functions. So because these are microbes, these are metabolic functions. And one nice figure. This is Metacoder. Our package is called Metacoder for making this, and it's it's quite difficult to use. It's very complicated, very complex data structure used for this package. So I've often given up trying to use it, but in this case I had some luck and managed to get it working. So it's a very nice taxonomic tree of comparing differences between microbiomes. A desata, A laboria, so these are honeybee species, incidentally. A stands for Apis, honeybee genus. General linear model, relatively simple statistical test. Some graphs of alpha diversity, again relatively simple. Linear models. These are using principal components. They will A, and then pollen phylogenetic diversity, human footprint, principal components, and their effects on microbiome. Alpha diversity table. So this is MRM. This is called. This is it's quite an interesting approach. This is for looking at beta diversity, and it's not too difficult to run, but pretty cool method. I like doing this, and it accounts for geographic distance when looking at. There's a more advanced method to do that, but this is a lot easier to do. And that's it. That's the main figures. Here's a study site. This is called Yunnan province in South China. This is a very biodiverse region, a global hotspot. But that's been characterized on plant diversity. A lot of the higher trophic groups are very poorly described, and so a lot of work needs doing. But for plants, botanists have done a great job of mapping plant diversity. But here these are the sites we went to collect bee nests. So you can see the bee species here, and is this honey or pollen which sample, and just some codes. Apis laboriosa, this is quite an interesting species. It's not the honeybee you'll see flying around cities. You have to go right into zoom into his picture here, right into a cliff face to find these nests. Very, very difficult to sample these. Apis cerana, you see this everywhere in Asia. Apis fluviat, and dosata, another quite different. Well, this is a larger honeybee. You won't see this around the cities, but what else we've got? This is error modelling. Quality score is very high quality for this microbiome, so that's why it's just some simple plot. And okay, some counts, and here functionality. So I was interested to do this because in plants and animals this is not really done with. It's very difficult to find people who have profiled functionality using using DNA barcodes in insects. But this is done in microbes, so I was interested to go through this process. Anyway, today, we'll very quickly whiz through some of the code and see how this was done. So .2, this is our library for processing metabarcodes or metagenomes. So all these commands, these are standard. You'll find that go to the .2 manual and tutorials. You'll find all these lists of commands. So this creates ASVs, which are high resolution entities from 16S metabarcodes. Okay, and that's it. Functional profiling, as I say, I was interested to look into this because I'd like to start applying this to insect data. But there's a bunch of methods, and I tried a lot of these, and most of them I gave up. Very difficult to use. One particularly was very easy to use, and this is what I would recommend. It's called Tax for Fun 2, and it's easy mainly because it is just a single dependency, and that is BLAST plus, which every bioinformatician would have this installed anyway, BLAST plus. So very easy to get this is running. So I would recommend, if you're doing functional profiling of microbiomes, you try that. And so you just need the KEGG database and some taxonomic 16S traditional database. And it outputs predictions of the pathways and the functional prediction from the KO system, which I'm not particularly familiar with. It's a microbiome metabolic function system. But I just simply integrate input that then into the standard alpha and beta diversity tests as an alternative to species counts or ISV counts or whatever. Okay, so yeah, this is this type of functional information you'll get from that method. So you pull out from this the more relevant things. So some of these are not relevant to, say, a nest microbiome. You wouldn't. Some of these are like a human disease related and would be. And skip ahead. Motor. This is for taxonomic classification via 16S reference database. Metacoder. As I say, this will give you very marvelous figures. Like this, but it's can be very awkward to run, very complex. So metacoder, if you're trying to do this is a single figure, like this is a taxonomic composition for a single sample, it's not too bad. But if you're doing multiple samples, then it's very difficult to make the data structure to do this, and sometimes very difficult. Let's continue. Some of the libraries used in our analysis. Environmental data. This I produced in a separate study for these same sites, and I use public environmental data and just which is in raster format, and pull out the variables for each site, like a human disturbance and forest cover, this kind of thing. But it's not processed in this project, so I can't show you at the moment how to do this. Reading the microbiome taxonomy and functionality created above. There's always various standardization. So for instance, the rows and the columns of your various tables, you use the intersect command to standardize these. And so you use intersect to get the overlapping names all ordered in a single way, and then reorder the tables so they're all equivalent. Now, when you have these metabarcodes, you need to do read normalization. Here's a package to do this, and commands you can find online. Some awkward process I had to go through here. I don't remember what this is for. Calculation of alpha diversity, CESPD. This is standardized phylogenetic diversity and various measures of alpha diversity from the functions and from the composition. And taxonomic diversity here, which requires this command using distances, and then taxondiv command gives you taxonomic diversity. And beta diversity, which is differences between sites rather than differences in numbers. And yeah, I'll just show this. This is so I did a printable component, which is not particularly, I don't have particular expertise on this, but I did this anyway. And so instead of using the primary environmental variables, which some of them will be strongly correlated, you do this printable component and get the first several of these, and then just instead of using a direct variable, you test the principal components of these, because you can see how many. These are all different environmental variables for climate, which is temperature and precipitation, forest variables, human footprint, and bioclimate. There's the box plots. Drop levels, that's when you need to. This is a general complaint I'd have about using R. It's you very much have to specify what is going to be in a variable, and which is very different from Perl, where you just put anything you want in there. And so even if you specify, you have different, say 3 different classes of some variable, and then actually only have 2 classes of that variable, even that can throw an error message, resulting in you have to use this command. Anyway, general linear model. Alpha diversity figures. On to beta diversity. And here's the MRM, which I don't remember exactly what, maybe matrix representation something, I'm not sure what. This is a way of looking at beta diversity and accounting for geographic distance. And you need an ecodist or package to do this, but it's pretty easy to run. There's a much more advanced version. I forget the name. And just plotting the map. Here's the map of Yunnan, and so he uses shape files. I started using shape files a few years ago. Reading the hillshade, but in the end I didn't bother using that. So in R, open a plotting device, plot the Yunnan shape, province boundaries, although most of these are outside the range of the plotting area, plot a map grid, which is these things. Then plot the text, so these are in order, and where the lines and the points on the map for these. And metacoder again is rather awkward to do this. In this case it worked, but I've failed to do this in other projects. And that's it. Okay, that's okay. Quick review of what was required to publish this paper, and go and check it out on your favorite publication repository, or you can get a copy off ResearchGate. Okay, that's all for today. Until next time.